Let us move inside and we will show you the R&D facilities. This is the School of Biotech Science R&D facilities. We start with the Molecular Genetics Lab. This Molecular Genetics Lab, as I have explained you, it's a DBT project lab. Let us come to the Molecular Genetics Laboratory where we have ongoing projects. The first ongoing project is the development of a RNA interference based silencing approach. We have received a grant of 55 lakhs and that grant includes ICR, Central Institute of Freshwater Aquaculture, and also in this particular grant we have 2 junior research fellows. Okay, let us come to the first system what we have in the Molecular Genetics Laboratory. We have the Applied Biosystems Quant Studio 3. This is the real-time PCR which we have procured last year, and in this particular instrument under our project we are going to study the gene expression. Let us move to the other instruments what we have in the Molecular Genetics Laboratory, starting off with a deep freezer minus 40 degrees centigrade. Now here we are keeping all the RNA and the cDNAs and we are storing it in this particular freezer. This also has come from the DBT project. And we also have a conventional PCR. The conventional PCR is used for the gene expression studies. Particularly, we are every day utilizing this particular system to do a semi-quantitative PCR of lectin transcripts. Now we have the normal refrigerator 4 degrees centigrade and also minus 20 degrees centigrade. And here we are utilizing this for storing the reagents and other purpose, and it is a very important asset for this particular laboratory. Coming back to the other instruments, we have the microwave oven, we also have the water bath, we have the UV transilluminator, and also the agarose gel electrophoresis system, which basically always is required when we isolate DNA and we run it on a gel to find out the integrity of the particular DNA. Let me actually tell you about one of the collaborative or consulting projects we are having with South Korea. We are having a consulting project with Sunchunyang University, which is in Asan. Under that particular projects, we are trying to study the endangered species, particularly the genomics and the transcriptomics approaches. Now, we are actually doing the downstream annotation part, following which we also have the other basic equipments like the micropipette, also a pH meter. We also have the gel rocker over here. We also have the Western blot apparatus, and we have many of them which is used for blotting particular specific immune proteins, and then we are going to study their immunological role using this particular system. Moving on, we also have the microscopes. We are using it not only in the cell culture and also in other experiments. We also have a UV visible spectrophotometer, and we also have a refrigerated centrifuge, which is microprocessor based centrifuge. This lab boasts of many chemicals and reagents, and that has been procured and that is all tabled in the stock register. We are into the microbiology facility over here, and as Trident intramural funded research is a very important component of Trident research apart from the extramural funding, we also have faculties, assistant professors who have taken up their own projects and they are working under this Trident intramural funded research. Basically, we would like to show you the laminar airflow where they are now working. The laminar airflow actually was a part of the Trident intramural funded research, and we are basically using this for that purpose. Also, we are trying to utilize the molecular genetics program here because we are trying to clone the necessary genes of interest and grow it in E. coli culture. So, in the microbiology facility, we also are supplementing our work with a orbital shaker. Let us move to a separate facility, the biocomputing lab. We are inside the biocomputing lab where we have close to 30 computer terminals with Windows and Linux operating system. We are using many softwares. These softwares are free in the public domain, and also there are some softwares which can be purchased. So we are using LaserGene, we are also using Blast2Go, we are using softwares like the sequence analysis software and the structure analysis softwares. We are moving to the second facility of research, R&D facility at Trident Academy of Creative Technology, that is the Cellular Immunology Lab. Now this lab came into existence with a transnational funded proposal which was accepted by the European Union and also the Indian counterpart, that is Department of Biotechnology, Government of India. So let us move into the Cellular Immunology Lab. The Cellular Immunology Lab is headed by Dr. Arup Sarkar. Dr. Arup Sarkar is my colleague and he has come from USA with extensive experience in cellular immunology. This particular project, he has many partners. Actually, he has 4 partners, European Union partners, and we also have adjunct faculty, Dr. Bhaskar Saha from NCCS Pune, who apparently comes over here to take care of the immunology program. BOD incubator. Now, particularly the Leishmania culture is stored over here. Now we are moving to the sterile room. So we are inside the culture room of this particular lab, and we could see research scholars are working. This particular project now houses another JRF who is now in collaboration with our adjunct faculty, Dr. Bhaskar Saha, is researching at NCCS Pune. Regarding the instrument, inside this culture room, you could possibly see the biosafety cabinet where Deepti and Sushri is working, and they are, I hope so, they are culturing the Leishmania over here. And we also have the centrifuge, the cell culture centrifuge, which was also been procured under this particular grant. We also could see Vasundhara and Tejaswini, who are working with the microscope over here, and it is attached to the particular software is attached to a particular terminal, computer terminal. And also, which is a very important aspect, is the CO2 incubator has been procured for this particular project. It was my pleasure to make you walk throughout this video of our R&D facilities. The library facilities include the journals, the competition refreshers, and other books which our researchers are utilizing for their studies and for their research work. We also have the e-library facilities which we are helping them to access through our computer terminals. The supporting labs include the bioinstrumentation lab, it's a central facility. We also have the microbiology, tissue culture, molecular biology labs where students are doing their practicals, and also the researchers have access to the equipments over there. One point of time, about 350 biotechnology students at the undergraduate level and about 50 to 60 MSc biotechnology students are studying. Thank you for having patience to watch this walk-in video. Thank you.